Welcome to Chapter 4, Part 3, Inbreeding, another look at pedigrees. So if we take a look at this particular pedigree, keeping in mind that pedigrees are a way to illustrate how genetic traits are transmitted through a particular family, through different generations, what we see is that this particular pedigree is the result of inbreeding or consanguineous matings. Now, these types of matings can reveal recessive traits because you find an increased incidence of homozygous recessive individuals when inbreeding is occurring. And what we notice is that all of these affected individuals here are the result of these consanguineous matings, which are denoted here by these double lines. Now, this type of inbreeding is not common. It's very rare in humans, but very common in laboratory organisms, whether it's plants or animals. Not only laboratory, but also in livestock. And the reason in terms of for laboratory animals or organisms is because you want a genetically pure inbred line so that you can eliminate any experimental variables that might be due to genetic differences between the individuals. The problem is that these inbred lines are typically less vigorous, which means they're sort of less healthy, and this is known as inbreeding depression. So we can see inbreeding depression in these varieties of corn. Okay, and these inbred strains, as I've mentioned or alluded to before in this previous slide, they're homozygous for alleles that are present in the founding or in the parental strain. And what's going to happen is that when you cross two inbred strains, so this one and this one, what you find is that the heterozygotes, or the hybrids shown here and here, are going to have what's called hybrid vigor. They're going to be more vigorous. That's also known as heterosis. And so when you're planting your vegetable garden and you look for seeds and you see on the seed packet that it says hybrid, that's what you want. They're going to be more vigorous plants. All right, so let's do some genetic analysis here. We've done genetic analysis before with mono and dihybrid traits. Well, now we're going to, or crosses, now we're going to do a genetic analysis of an inbred individual, I, who has parents AB, and AB are half-siblings. Now, I is inbred because I is the offspring of these half-siblings. So A and B share a father, but they have different mothers. Now, because I receives one copy of each gene from A and one copy of each gene from B, and these two parents here also receive their DNA, their chromosomes, from individual C, it's possible for individual C, lost my cursor, individual C to have the same genes as individual I. And if that happens, we say that those genes are identical by descent. Now, the question is, what are the chances, what's the probability that I and C can have identical gene copies, and so would be identical by that the gene copies would be identical by descent? Well, first we have to we can calculate a coefficient that allows us to determine what that probability might be. But first we have to find something called an inbreeding loop. That'll become clear in just a moment, but let me just explain the inbreeding loop. So the inbreeding loop essentially is the path that the genes can take from the common ancestor here of the inbred individual and to the inbred individual. So this is one inbreeding loop here, and it consists of three individuals. 1, 2, 3. In this case, we do not count the inbred individual himself or herself in this in the inbreeding loop. All right, so the inbreeding coefficient, as I said, is essentially the probability that two gene copies in an individual, and an inbred individual, are identical by descent from a common ancestor. So they're identical to those gene copies you would find in that common ancestor. So to calculate that coefficient, abbreviated with F, first you identify the common ancestor of the inbred individual, then you count the number of individuals in each inbreeding loop, which I just showed you on the previous slide. Then you calculate this quantity, 1/2 to the nth power, n meaning the number of individuals in the inbreeding loop. You do that for each inbreeding loop, and then you add up the results for each loop. All right, so as an example, let's calculate the inbreeding coefficient for this offspring here of a full sibling mating. So R and S are full siblings. U and V are the common ancestors. Because there are two common ancestors of Z, there are two inbreeding loops. Okay, so now we have to count the number of individuals in each inbreeding loop. All right, so for loop 1, This is two loops, actually, and they each have, remember, there's two loops because there's two common ancestors. There's three individuals. 1, 2, 3. Remember, we don't count the inbred individual. 1, 2, 3. Okay, now we calculate this quantity for each inbreeding loop and add the results. So loop 1, we've got 1/2 to the third power, which is 1/8. Same for loop 2, add them together. Your inbreeding coefficient for full sibling mating is 1/16, which means that Z has a 1 in 16 chance of getting the exact same gene copies from U or V due to this concept of identical by descent. All right, but what if the ancestor himself is inbred? Well, that's a different calculation. You have to multiply your coefficient of your inbreeding coefficient by the sum of 1 plus the same coefficient for the common ancestor. Okay, that sounds a little complicated. Let's do an example. Here's your common ancestor. Let's first calculate the inbreeding coefficient. This common ancestor has one common ancestor here, so one inbreeding loop, 3 individuals, 1, 2, 3. So that coefficient is going to be 1/8. Now, to calculate the inbreeding coefficient for individual T, here's our one common ancestor, one inbreeding loop, 1, 2, 3 individuals. So the inbreeding coefficient for individual T is going to be 1/2 to the third power times 1 plus the coefficient for the common ancestor, for a total of 9/64. Okay, so why do we even want to calculate this inbreeding coefficient? What are the uses of this? Well, we can use it for a couple of different reasons. One is to explain the increase in the recessive conditions that we observe in these consanguineous matings. So basically to explain why we see so many homozygous individuals with these homozygous recessive conditions by consanguineous matings when we look at those pedigrees. Secondly, we can use it to help explain the decline in vigor, essentially, in inbred strains. So essentially what you're looking at here is plant height for an inbred strain, crop yield for an inbred strain. Essentially, as the inbreeding increases, as the inbreeding coefficient increases, you find that the crop yield declines and the plant height declines. So what this allows us to conclude is that the accumulation of maybe harmful, maybe not harmful, but essentially the accumulation of homozygous recessive traits is the cause of this inbreeding depression that we see in these inbred lines. Okay, so something related to inbreeding coefficients is, what about measuring the genetic relationships between within families? How close are certain genetic relationships? So if we think about an uncle-nephew versus a half-sibling relationship, which of this relationship is more closely related? And we can calculate this, so we can look at it objectively, and we can do that by calculating something called the coefficient of relationship. It's the fraction of genes that two individuals share simply by having a common ancestor. Now keep in mind, these are not inbred or consanguineous matings situations. So the way that we calculate this is we take the predicted inbreeding coefficient, so we essentially calculate that inbreeding coefficient for some imaginary inbred siblings, and multiply it by 2. Very simple. So if we were looking at full siblings, the coefficient of inbreeding, the inbreeding coefficient for an imaginary full sibling would be 1/4, and you can calculate that out using following the examples we did in the past few slides. The coefficient of relationship would therefore be 1/4 times 2, which would be 1/2. And so what that tells us is that full siblings share half their genes, which makes sense because they each get half of their genes from each parent. This concludes part 3 of chapter 4.